Hello students. In the last class, when I talked with you regarding the topic photosynthesis, I got to know that many of you are facing problem in internet connectivity. So, I have arranged this one. To clarify your doubts, to clear your concept, here I will give a brief idea on this photosynthesis topic based upon your class 11 standard. Let me clear you first. A class can never be replaced by this kind of videos. But this will help you to clear the idea what you are getting from the class. As always, I need your attention properly. So let's start. We are focusing in the photosynthesis. That is, of course, this photoautotrophic part of the nutrition. What is the topic? Photosynthesis means a physiological process by which green plants synthesize carbohydrate with the help of chlorophyll in the presence of sunlight, taking CO2 and water from air and soil respectively, and with the release of oxygen and H2O as byproducts. So hopefully no need to explain this part more. This diagram is quite well known to you, as already you have gone through it in class 10. We are focusing here, the stoma part, where from they are taking the air, or the transport is done regarding the air, CO2 and oxygen. After that, we are getting the chloroplast also, their base photosynthesis part. This is a plant cell where the chloroplast, a double membrane bound cell organelle is present, within which the granum is present, within that the thylakoid, the first part of the photosynthesis. In brief, we can see the chlorophyll present in the thylakoid, green coloration, captures energy and forms ATP, adenosine triphosphate. In the right side, in blue color, ADP combining with the inorganic phosphate and giving the ATP, right? That's the light energy to chemical energy conversion. The second most important, to supply the electron, the continuous source, as the electrons after taking sunlight excited and then coming to its original position. This is the key concept. Okay, chlorophyll, a molecule which is having magnesium in its center. Other things are carbon, hydrogen, oxygen, nitrogen. Here from electrons are excited after getting the photon particles from the sunlight. Electrons are going above. After some time, it is again coming to its original position. So to supply the electrons, we need the splitting of the water molecules. And after splitting the water, H plus is going to reduce the NADP, is going to be reduced to form the NADPH. So this ADP plus Pi means ATP and NADPH are the assimilatory power which we need for the Kelvin cycle. Reduced NADP with the help of enzymes combine CO2 and a 5-carbon component to make 6-carbon compound. Glucose is formed here. This side we name it light reaction in grana. This side dark reaction in the stroma of the chloroplast. Now, as per your class 10 standard, these 3 lines you got to know. Going in the deeper concept, this is the electromagnetic spectrum. We are seeing the visible light. Okay, and where from, if we are focusing we are getting the red colored light about 650 to 780 nanometer, and blue colored spectrum 400 to 445. This part we are getting two new terms, absorption spectrum and the action spectrum. What is absorption spectrum? A curve obtained by plotting the amount of absorption of different wavelengths of light by a particular pigment. That is the absorption spectrum. Here we are talking about two main chlorophyll pigments, chlorophyll A and B. So we are seeing when we are plotting the amount of absorption of different wavelengths, that particular pigment, we are getting absorption spectrum specially for the blue and the red color lights. And the curve showing the rate of photosynthesis at different wavelengths of light is action spectrum. So when we are going to show the rate of photosynthesis at different wavelengths of light is the action spectrum. Both this absorption spectrum and action spectrum that curve in your NCERT text, go through that. This is the part where we are getting the PS1, PS2 together, figure Z scheme first, and then the cyclic phosphorylation. We are focusing here. This is the LHC, the light harvesting complex, where P700 is the reaction center. In any this kind of system, LHC, light harvesting complex, one will be reaction center, maybe chlorophyll A or B. Apart from that, there will be the antenna molecules, core molecules. Antenna molecules absorb light of various wavelengths. Okay, then after that pass it to core molecules and ultimately to the reaction center. Here we are seeing light is falling, chlorophylls are excited, then the electrons are carried by few electron acceptors. Ultimately, electrons are again coming back to the original position. So that is photosystem 1, where ATP can be formed by getting the energy from those released electrons. So this is also a kind of phosphorylation, photophosphorylation. Okay, but this is cyclic photophosphorylation, and this one here we are getting the non-cyclic photophosphorylation, where the reaction center is P680. Chlorophyll going to absorb 680 nanometer light. Ultimately, chlorophyll is excited, electrons excited. They are having few electron acceptor. When electron is going to the lower orbit, energy is utilized to form the ATP. And those electrons again can be reutilized in the photosystem 1. So here photosystem 1 and 2 both are working together. This is the non-cyclic scheme or the Z scheme, where NADP is also reduced. Let me clarify, both the things are important, cyclic and non-cyclic. Especially non-cyclic one, we are getting more. And most important, because here only the photolysis of water done. So oxygen, which is the important byproduct of the photosynthesis, we are getting from here only, not from the non-cyclic one. From the non-cyclic one, we are getting the oxygen. And here from only we are getting the NADPH also, important to reduce the CO2. This is the beta diagram. Who wants the names of the carriers in cyclic and non-cyclic, both many carriers names are given, like plastoquinone, pyridoxine. Any iron sulfur protein, there will be cytochrome complex, there will be plastoquinone, plastocyanin, this. Okay, so moving on, ultimately what we got assimilatory power, ATP and NADPH, which we need to reutilize in the Kelvin cycle. Cyclic and non-cyclic phosphorylation difference. Only PS1 is involved in cyclic. Non-cycling, both are involved. In cyclic, no photolysis of water. In non-cyclic, photolysis of water. In cyclic, oxygen not evolved, whereas in non-cyclic, oxygen evolved. In cyclic, NADPH plus H plus is not synthesized. Non-cyclic, it is synthesized. So here, in cyclic, electron is cycled back. Non-cyclic, it is not done. Moving on, photosystem 1 and 2. You can go through all the differences. I am focusing in the main part. Photolysis of water is not occurred in non-cyclic. Here reaction center is 700 and molecular oxygen is not evolved. Opposite things are in photosystem 2. So already we have a clear idea regarding the first half, whatever happening in the grana known as the light reaction. Moving on to the ATP synthesis through chemiosmosis. Whenever we are talking about water hydrolysis, H plus is going to be more in the inner side of the thylakoid, that's the lumen. We can see here this chemiosmotic theory in 1960s. Peter Michel proposed this theory, where due to photochemical reaction inside the thylakoid membrane, H plus concentration is higher than the stroma. So according to this theory, H plus enters the stroma side through CF0CF1 ATPase due to proton motive force PMF. Already I discussed in class. PMF, proton motive force, and ADP and PI utilize this proton motive force to produce ATP through ATPase, which is bringing a conformational change to the ATPase. In ATP synthase, F0 is the base, F1 is the head. Moving on to the next part. We have seen here carbon fixation, then reduction, and then regeneration of the RUBP. Moving back to the C4. C3 we understood. Here the first product CO2 with the help of the Rubisco enzyme in presence of RUBP, they are forming the first three components, three carbon component name is PGA, phosphoglyceric acid. It's a 3 carbon components. Whereas when we are talking about C4, there the first component is the, we can see CO2 is coming. In the mesophyll phase, in presence of carbonic anhydrase, oxaloacetate is formed. How? This one carbon is going to be bound with PEP, phosphoenol pyruvate, which is coming from the pyruvate in presence of PEP carboxylase. Once OAA, oxaloacetate form, it can be converted to malate, it can be converted to the aspartate, whatever the thing, ultimately it is coming to the bundle sheath cell, which is having few extra kind of the feature. What are the extra kind of the features? In case of the bundle sheath cell, they are having the Kranz anatomy. Here, the vascular bundles are surrounded by outer mesophyll, inner bundle sheath cell. And in bundle sheath chloroplast, we can see these are small, do not contain starch and have well developed grana. So in bundle sheath chloroplast, we can see compared to the mesophyll chloroplast, as mesophyll chloroplast are small, do not contain starch, have well developed grana, and in bundle sheath chloroplast they are large, contain starch and are either agranal or rudimentary grana. Remember this point, that bundle sheath chloroplast are either rudimental granule or they are agranal. In this way, here we can see they are utilizing the malate from the oxaloacetate in the malate, and they can again release the CO in presence of the Rubisco. They can again enter the Kelvin cycle. What is the benefit of this one? The benefit is a C3 plant CO2 acceptor is the RUB. Here the CO2 acceptor is the PEP, and it operates a faster rate of the CO2 fixation in C4, faster rate of CO2 fixation, and photorespiration is absent, means utilization of CO2 is maximum. They can increase the pressure of CO2 within the cell. That's the main important thing. So let's see this mesophyll cell, your book diagram, phosphoenol pyruvate, C4 pathway started. Again it's coming through this point, plasmodesmata, the cell joining to the bundle sheath cell, where they can again enter the building cycle. Photosynthesis occurs in mesophyll tissue. In C4, photosynthesis occurs both in mesophyll and bundle sheath. Kranz anatomy absent, present. Kranz spelling is wrong here, okay? Here the optimum temperature 20 to 25, here optimum temperature 35 to 44, means in the tropical climate. And most important, photorespiratory loss, which is negative, which is not good for the plant, is high. Here the photorespiration does not take place. So what is photorespiration? This is the C3, C4 and CAM. CAMO I will discuss in class only. This is photorespiration, where we can see RUBP can be reutilized both with the CO2 and oxygen in presence of Rubisco. If Rubisco enzyme is getting oxygen, it can even bind with oxygen. That's a wasteful process. Here we can see in chloroplast, Rubisco can again bind with the oxygen to form the glycolic acid, 3-phosphoglyceric acid and 2-phosphoglycolic acid. This glycolic acid or phosphoglycolic acid is not good for the plants in long run, because photorespiration does not produce ATP, rather it consumes energy. During the photorespiration, ATP and NADH both are consumed. And during photorespiration, there will be 25% loss of fixed CO2, which plant absorbed at the time of photosynthesis. Therefore, photorespiration is wasteful method. Main thing, Rubisco is engaged here to bind with the oxygen rather to bind with the CO2. Peroxisome, mitochondria, chloroplast are involved. Ultimate thing is oxygen utilized and ATP utilized, but no net product, no formation of glucose. This is done in C3 plant, not done in C4 plant, as they have already increased the concentration of CO2. So Rubisco is now going to bind with the CO2, not with the oxygen. Rubisco is, we can say, an opportunistic enzyme. More oxygen pressure, it will bind with oxygen. More CO2 pressure, it will bind with CO2. As here, C4 plants are increasing the content of the CO2. So, Rubisco is not going to perform this photorespiration. That's it. Brief. Thank you. In class, you can ask about your doubts. Thank you all.